All right, so in this chapter we're going to be looking at blood, and so we'll start with some introductory information, functions, and so on of blood, and then we will talk about red blood cells, the erythrocytes. So when we think about the functions of blood, transportation of materials, of nutrients, of gases, cell waste, hormones, movement of stuff around the body is a big, important primary function. And also, there is a lot of communication between the fluids of blood and the fluids of the rest of the body. And so, blood also helps to maintain homeostasis in the interstitial fluids of the body. And remember, interstitial fluids are the fluids that are found outside of cells, but between the cells. So in interstices, those are like the cracks in between things. And so interstitial fluid is the fluid that's around the cells, because all of our cells, in order to maintain or remain alive, must be bathed in the fluids of the body. As well, we produce heat in organs like our muscles and the liver, and that heat must be distributed around the body so that the body can continue to function properly. So distributing heat, and then some of the formed elements of the blood are involved in immune function, and so protection from disease is another important function that the blood provides for the body. So there you got it. Lots of functions associated with the blood. We're not going to spend any time in this chapter focusing on disease prevention or protection from disease, immune system function. That we're gonna leap into that 2 chapters from now when we talk about the lymphatic system. Okay, so blood is a connective tissue, and it's been a while since we've talked about connective tissues, so I want to remind you of a couple of things about connective tissues. When we talked about the components or things that made connective tissues, how we could identify connective tissues, there were 3 things that we talked about. Specialized cells are found in connective tissues, and we're going to see specialized cells as we talk about blood. And then external to the cells is a material referred to as extracellular matrix, and that extracellular matrix is composed of two pieces of stuff. There's the ground substance, and that is the fluid or the gel that is around the specialized cells. And then proteins, and in most connective tissues, the proteins are fibrous. There's some sort of protein fiber, and we talked a lot about the protein fibers. They're elastic fibers and reticular fibers and collagenous fibers. Those are the 3 primary fiber types. And so when we talk connective tissues, it's specialized cells plus the extracellular matrix. So blood, excuse me, oh man, goodness, there we are. I think that's a little better. So blood, how is blood a connective tissue? Because we don't see this firm, cartilage-like extracellular matrix. We don't see the kind of strappy, very tough extracellular matrix that we see, say, in a ligament or a tendon. So how does blood fit in as a connective tissue? Well, specialized cells certainly we have. There are erythrocytes. Erythro, by the way, means red, and cyte, of course, means blood, cell. So these are the red cells that are found in the blood. Red blood cells, and if you're taking notes, boy, I would go with an abbreviation like RBC for red blood cells. So red blood cells, their function is to transport gases, oxygen and carbon dioxide specifically. And here's a red blood cell. And leukocytes, these are the white blood cells. Leuko means white. So And again, if you're looking for an abbreviation, WBC is what I would use. White blood cells are the ones that are involved in immune function and in cleaning up, removing damaged tissues. And here are some examples, some images. They are stained, so this coloration that we see here is not the real coloration that you would see in a white blood cell. They are whitish colored, but the graininess is what they're trying to demonstrate for you by staining them. So these are some examples of white blood cells. So those are the specialized cells. The extracellular matrix in blood is blood plasma. So plasma is primarily water, about 92% water, and the rest of it, the other 8%, is almost all dissolved proteins. And then there are some other things as well, but the bulk of the 8% left over are dissolved proteins. So, let's see. So some fun facts about blood. Woo! Ain't nothing but a party when it's come times to talk anatomy and physiology. In the average person, there's about 5 liters of blood. A liter is slightly more than a quart, so we're looking at somewhere between a gallon and a quarter and a gallon and a half of blood in the average person. And the amount of blood that we have in our bodies varies, of course, with body size. Larger people have more blood, but it can also be influenced by electrolyte concentrations or fluid concentrations in the body. So if you were to eat that country ham at Easter time, then that ham that was cured in salt, then you're going to be adding a lot of electrolytes to your body, and in response to that, you're going to become thirsty and want to drink more fluids. And so the volume, as you add salt to your blood, water is going to diffuse into the blood, and so the volume of blood is going to increase. So increase in electrolytes equals an increase in blood volume, which then results in, well, think for a minute, what would that do to blood pressure? Hopefully you just said, well, it's going to raise blood pressure. So adding electrolytes will add fluid to the blood. If we lose fluids, let's say you are working outside on a hot, sweaty day, then you're going to lose fluid from your blood and blood volume would decrease. And then more adipose tissue equals more blood, because adipose tissue requires lots and lots of blood supply, and so as you're increasing the capillaries serving adipose tissue, then you're certainly going to increase the blood volume. Okay, so hematocrit is one of the things that we can measure about blood. So hematocrit, abbreviated HCT, sometimes called packed cell volume, is the percentage of your blood that is made up by the red blood cells. So we can measure hematocrit, so sort of the gold standard for measuring hematocrit would be to fill up a 10 centimeter, is that right? About a 10 centimeter capillary tube. That's a very thin glass tube, and blood gets drawn up into it by a physical property of fluids called capillary action. And so it's just automatically drawn up. The blood, you poke a hole in you draw blood up into that capillary tube, and then you toss the capillary tube, I say toss, you place it carefully into a centrifuge and spin it round and round and round and round and round for about, 5 minutes. And the spinning around is going to force the blood to separate into fractions based on the density of those fractions. And so we're going to drive the heaviest red blood cells toward the bottom of the capillary tube by spinning it in that centrifuge. And then you can measure. If you have 10 centimeters of blood and we measure that this is something like 4.5 centimeters, then that would tell us that we have approximately 45% red blood cells. And then the remainder is is blood plasma and white blood cells. So the average for humans is about 45% red blood cells, but this is different for men and women. So men range between 45 and 54% red blood cells, while women have a slightly lower, 37 to 43% red blood cells in their blood. And the primary reason for this difference between men and women is all about the reason that, the difference between men and women. It's testosterone. Testosterone is a great stimulant to the kidneys, telling them to make a hormone called erythropoietin. Anytime we see poi, that what I think is production. So erythropoietin is a hormone that is produced by the kidneys in response to low blood oxygen. And when the kidneys notice that, hey, blood oxygen is getting a little low, they produce erythropoietin, and that tells the red bone marrow, hey, it's time for us to kick up the production of red blood cells. So because testosterone is a big, it's like a big old push to the gas pedal for erythropoietin production, then men produce more erythropoietin just as a baseline, and so we have higher red blood cell counts than women do. There is a very thin layer when you do this centrifuging. There's a very thin layer, sometimes referred to as the Buffy coat, and it is where the white blood cells and these little cell fragments that are referred to as platelets are found. So we'll talk more about white blood cells and platelets in a later lecture. And then the remainder, about 55% of blood volume, is the plasma, the water and the dissolved proteins that are found in the water of the blood. So hematocrit is the amount of red blood cells that are found in the blood. Hematopoiesis, so here's another little definition for you. Hematopoiesis is the process of producing blood. This prefix hema means blood, so this is blood production or blood cell formation. Hematopoiesis is done in red bone marrow, and it's done by a blood cell called hematopoietic stem cells, or sometimes they're called hemocytoblasts. So blasts, recall, blasts, we took that to mean builder, so these are the blood cell builders, or hematopoietic stem cell, that refers to blood production stem cells. Stem cells, if you recall from our conversation about that last semester, these are cells that are partially differentiated, and when they divide, and they're capable of continuous division, and when they divide, their offspring cell, one of the daughter cells, is a specialized cell, and the other daughter cell remains a hematopoietic stem cell. So if you find this drawing in your book, look for this one, and you will be able to read the words a lot better than what we can read in this slide. Wrong way. There, I can make it bigger. But here's our hematopoietic stem cell, and now I can't draw on it, so that's one of the downsides. I make it bigger and then I can't draw. But our hematopoietic stem cell, as it divides, can produce myeloid stem cells or lymphoid stem cells. So under erythropoietin, the hormone, these hematopoietic stem cells divide and they produce myeloid stem cells, and those myeloid stem cells will then grow and become either any of these four cells: proerythroblasts, megakaryoblasts, myeloblasts, and these three here are myeloblasts or a monoblast. So with erythropoietin around, that myeloid stem cell will divide and become one of the offspring cells will become a pro-erythroblast, and that will send then that cell down this developmental timeline. So the pro-erythroblast will mature into an early erythroblast, which will mature into an intermediate erythroblast, which will mature into a late erythroblast. Notice something here. Look at what it doesn't have inside the cell anymore. We've said goodbye to the nucleus. The late erythroblast will mature into a reticulocyte that gets released into circulation, and it will mature into an erythrocyte, where it is now functioning as a mature red blood cell. I'm going to make that small again. Now I can write on it. Okay, so hematopoietic stem cells, they're capable of dividing, and their daughter cells will grow into or develop into these more specialized blood cells. And with exposure to specific hematopoietic growth factors, and erythropoietin is one of those growth factors, then the development of the offspring cells are going to be sent down a particular developmental pathway. With erythropoietin, then what we see is the daughter cells are sent down this pathway, but the daughter cells could be, with other growth factors around, be sent into these other pathways as well. So, for instance, thrombopoietin, TPO, will stimulate the development to go down this pathway right here, producing a megakaryocyte, which is the cell that produces platelets. Okay, so that's hematopoiesis in general. We're going to stay focused on erythropoiesis. So red blood cells, again called erythrocytes, are very specialized cells. They have this shape. It's referred to as a biconcave disc because there's a concave surface here and a concave surface here. So that's why it's a biconcave disc. This biconcave disc has the function of increasing surface area. Let's say we have, for comparison, here's a non-biconcave, just regular disc, and we compare that to the biconcave disc. So which of these, if we sent a little ant crawling along this surface area, and the ants were moving at exactly the same speed, this ant A, and here's ant A, here's ant B, ant A would reach the destination here more quickly than ant B would, because the shortest distance between 2 points, here's a point and here's a point, is always going to be a straight line. So because this ant is following a slightly longer curve, then we can tell then that this shape increases surface area, this shape here, compared to this shape. So B has greater surface area than A does, is what I'm trying to say. And that got confusing, but hopefully you get what I'm saying here. So that means that the hemoglobin that is found inside, so now I'm drawing hemoglobin molecules, those hemoglobin molecules are all going to be closer to the membrane because of the biconcave disc shape. And that means diffusion is going to happen more quickly, and the hemoglobin molecules are going to pick up and release oxygen and carbon dioxide more quickly than they would if we didn't have this biconcave shape. It also has a second benefit. As cells pass through really, really narrow capillaries, the red blood cells sometimes have to bend and flex to make it around corners, and this shape causes a little bit more flexibility. So this thinner shape here in the center causes the cell to be a little weaker than it would be if it were a flat non-biconcave disc, and so it can flex more easily. Red blood cells have a lifespan of something approaching 4 months, about 120 days. As they get older, they become less flexible, and they stop flexing and tend to break. They become a little bit more brittle, and so that is the end of their life when they break like that. and they carry within them lots and lots and lots of molecules of a protein called hemoglobin. And so HB, that's what is it when you shorten a word? Anyway, that means that means hemoglobin. Gosh, maybe it's early in the morning and I'm having a hard time remembering stuff. It's the abbreviation. That's what I meant to say. It's the abbreviation for hemoglobin. Okay, so red blood cells, they have on their surface, so here we have the surface of a red blood cell. They have on their surface these identifier proteins called glycoproteins. It's a protein plus a sugar. These identifier proteins that give us what we refer to or think of as the different blood types. And so there are A identifier proteins, B identifier proteins, and RH identifier proteins. So we have 3 different proteins that can be found on the surface of a red blood cell that give us the blood types that we are going to be talking about at the very end of the chapter. As a red blood cell matures, it becomes anucleate. Anytime you see the prefix A, that means without. So an anucleate cell is one that does not have a nucleus. And so late in red blood cell development, they kick out their nucleus and they spend that 4 months circulating in the blood without a nucleus. That that's functional. It provides more space for hemoglobin to be found inside the red blood cell. But it also has a consequence. Because red blood cells don't have a nucleus, they can't repair if they get damaged. There's no instructions for making proteins, so there's no repair. And as they age, then they're removed by either the liver or the spleen. As they're making their way through those really thin little capillaries and they break up, then the liver and spleen get rid of them. As well, they don't have any mitochondria, so that means that they don't have any way, they don't have the enzymes required to make ATP using oxygen. So this is just one more means of ensuring that red blood cells aren't sipping on the oxygen they're supposed to be delivering out there to the body tissues. They still make ATP, they just don't do it with oxygen. So that's the deal. It's just that they have very low energy needs, and they don't do their red blood cell or their ATP production with oxygen. Erythropoiesis then is this process of producing red blood cells. It is done by negative feedback. So if the kidneys are low on oxygen, if the kidneys or the liver, to some extent the liver is involved here, but the kidneys are their primary EPO producers. If the kidneys are low on oxygen, so oxygen content is falling, and there's a number of things that can cause this low oxygen situation. It could be because you've lost blood. If you go to a higher altitude, the body notices this. It says, whoa, we don't have much oxygen. And so it will respond. Or if you're in exercise, doing a lot of exercise, the body responds to that as well. It notices low oxygen, and under those conditions, it will produce erythropoietin. So we've gotten low oxygen conditions, and in response, the red blood cells producing tissues of the red bone marrow are going to get engaged, and they're going to produce more red blood cells, which can carry more oxygen. And then we see a reversal. Remember, negative feedback says stop, go the opposite direction. So if O2 gets down, then we're going to see a reversal after some time, some exposure to EPO, and oxygen levels will climb again. So negative feedback is involved here. Erythropoietin can really put the gas pedal to red blood cell production, and under full EPO stimulation, then the body can be producing, and this is just a crazy number, between 2 and 3 million red blood cells every second. That is freaking fast. So just to maybe give an example of this, I spent the Christmas break, I think I probably told you, that I spent it in Bolivia visiting my daughter. We went hiking, and we found ourselves up at about 13,000 plus feet above sea level, which is pretty dang high. Oxygen content of the air is pretty dang low under that condition, and we were up there for quite a while, for 3 days or so, and I'd already been at 12,000 feet for, I don't know, 4 or 5 days at that point. I suspect that my body was really kicking it into high gear at that point. So I may have been somewhere around that 2 to 3 million red blood cells per second production at that point. So, to me, that's just crazy to think about that kind of cell production rates. Polycythemia, then, is the condition of excessive red blood cell production. This is when red blood cell production becomes pathological. So polycythemia, poly means many, cell in the blood is, so you can really just break down what do those terms mean. Polycythemia is excessive red blood cell production. It's an issue because that can increase the viscosity of blood and really slow down blood flow. Anemia, on the other hand, A, and then remember, is without. So here we are with low red blood cells in the blood, or perhaps we might have a normal number of red blood cells, but low hemoglobin content in those red blood cells is the condition known as anemia. And I've got some information in your notes that relates to the dietary factors and erythropoiesis. So B12 and folic acid, notice, are very important vitamins that support the production of red blood cells. Okay, a little bit here about hemoglobin now. Hemoglobin is about 1/3 of the volume of a red blood cell, and we have about 280 million hemoglobin molecules in a red blood cell. So it's a lot of hemoglobin that's found there. It's a globular protein, so that means that proteins are chains of amino acids. So we're going to go back and do a little chemistry here for a second. So it's a chain of amino acids. These are amino acids that have been linked together into long chains. The chains then will form helices, and sometimes they will form sheets that are kind of folded up like this. So what we're seeing here are these helices of amino acid chains, keeps going and then it bends around. So there are 4 of these chains of amino acids found in a hemoglobin molecule, and they kind of ball up, they fold up into a very specific shape, and those 4 chains then surround 1, 2, 3, and then here's the 4th of them, 4 heme groups that have at their core an iron molecule. So we have an iron ion there at their core, and it's this heme group that is capable of binding oxygen and then releasing oxygen. So in our lungs we pick up oxygen at the heme group, and then out at the body tissues we release the hemoglobin, release the oxygen. When we bind oxygen, it's called oxyhemoglobin. As we release oxygen, it's called deoxyhemoglobin. And then the protein part, that's what binds the CO2 molecules. So we pick up CO2 in our tissues, and we release CO2 in the lungs. When we pick up CO2, it's referred to as carbaminohemoglobin. That's hemoglobin with carbon dioxide bound to the protein portion. Okay, last slide then is about the destruction and recycling of red blood cells. So as we said, the lifespan of a red blood cell is about 120 days. the proteins in red blood cells can become damaged, and especially there in the membrane of the red blood cell. And if that happens, because there's no nucleus, there is no ability to repair the red blood cell or to replace the proteins that have become damaged, and so that's the end of the life of the red blood cell. It especially shows up as fragile cell membranes that can no longer bend and flex. And so when they get destroyed, or when they get damaged, they are broken down in the liver and the spleen, and then we recycle the. That's interesting. This should be a little bit more information rather than about polycythemia and anemia. It should be about how we recycle them. And I've got lots of notes in here for you to review about how the hemoglobin is broken down into its 4 globin chains and the heme group that's associated with it. The heme then can be further broken down so that we can recycle the iron, and then we produce a greenish pigment that's referred to as biliverdin. The biliverdin then is converted into bilirubin, and we get rid of both biliverdin and bilirubin as bile pigments or in the feces. So fecal material is another. It's produced into bile pigments that are used then to help assist with digestion in the blood, and so all those pigments are going to disappear as we poop out the waste materials. The iron is transferred, is attached to a transport protein called transferrin, and transferrin will take that back to the iron back to the red bone marrow so that we can produce more hemoglobin. And then those globin molecules are broken down into amino acids, and then we can reuse the amino acids. So we'll recycle those amino acids into other proteins. Okay, so that information should have been here on this slide. And just to add to the confusion, here's an image. This is a microscope view of what polycythemia would look like if you put red blood cells, a specific volume of blood under a microscope and looked at it. You would just see this huge clustering, a large amount of red blood cells. Here we see an iron deficiency in anemia. So these are low red blood cell count on this slide. So 2 different ways of looking at it. All right, that is it for this lecture. So any questions, you know what to do. Reach out to me. Send me an email. Give me a phone call. Find me during office